University, where he worked on crystallography and enzymology of carotenoid cleavage enzymes. He joined our lab just under 4 years ago, primarily to push forward structural biology efforts in the lab, and in particular, cryo-EM work in collaboration with Mahfoud Liao in the Harvard Medical School Cell Biology Department. And Xuewu has worked extensively on some of the membrane proteins involved in lipid droplet formation, including sapin, in which he's published a cryo-EM structure of the Drosophila enzyme. And he's going to tell you today about the DGAT1 enzyme that makes the triglycerides for the lumens of lipid droplets. And I'll just add that, you know, having been there at the very lab meeting where we discovered the DGAT1 enzyme in 1989, I never dreamed that we'd have molecular insight to this enzyme during my career. But Xuewu made it happen in our lab, and he's done some beautiful and outstanding work, so I'm quite excited for him to tell you about it today. Xuewu.
All right, let me share the screen. Okay, thanks Bob for nice introduction, and thanks for organizer for this wonderful event. So today I'd like to talk about our recent published work on the human DGAT1 structure, which is involved in the triacylglycerol synthesis. And again, this is a collaboration between Farese Walther lab and also Mahfoud Liao's lab in Harvard Medical School. I'm actually working in both labs as a joint postdoc. So as you might all know, triacylglycerol or TG is energy storage lipid molecule in eukaryotic cells. It contains a glycerol backbone conjugated with 3 fatty acyl chains with ester bond linkage. And the energy condensed in this highly reduced acyl chain can be oxidized to provide energy. Okay? And because this is a highly hydrophobic molecule, it cannot dissolve in the cell, in the aqueous environment. So when present in excess, cell developed an organelle called lipid droplet to store excess neutral lipids, including TG or other lipids like cholesterol ester and waxes. Okay, and the neutral lipid is stored in the core of lipid droplet. The surface is covered by a monolayer of phospholipids. So the biosynthesis of lipids of TG has been actually elucidated many years ago by Kennedy and colleagues. So now it's known as the Kennedy pathway. So the initial substrate, acyl-CoA and glycerol 3-phosphate, by adding this acyl chain from fatty acyl-CoA in a sequential manner by the enzyme, mostly at the ER or endoplasmic reticulum, finally generate DAG. Okay, and the final, because you know DAGs can be used for phospholipid biosynthesis, the last step to generate the TG is by DGAT enzyme or diacylglycerol O-isotransferase. Okay, and the hydrophobic TG product is believed to be released back to the ER lipid bilayer, which can be further used for lipid droplet formation. So as Bob mentioned, this enzyme was first cloned by Bob. And if you look into the reaction catalyzed by DGAT1, actually it's involving by the break of the cell ester bond in the fatty acyl-CoA, which is the acyl donor. And DGAT1 transfer this acyl chain to the OH group in the acyl acceptor. Here is the DAG, okay? And by reforming this ester bond to generate TG. And interestingly, beside a DAG, DGAT1 can also recognize other acyl acceptors, for example, long-chain alcohol as well as retinol to form waxes and retinol esters. So this the DGAT activity has been known for a long time, like involving TG metabolism, and its important physiological functions has been demonstrated everywhere. But we really want to understand, like, at the molecular level, how DGAT1 catalyze the formation of TG by capturing this semi-hydrophobic substrate and where to catalyze the reaction to generate this highly hydrophobic TG product, which is finally end up in the lipid droplet. We really want to understand behind the molecular mechanism. So to answer this, we are using structural biology and biochemistry method to tackle those questions. So we first express human DGAT1 and purify them in detergent. So after different trials, we finally reconstitute them back into amphipoles. As you can see, this is gel filtration. We definitely got two different species, the larger species and smaller species, okay? And we next utilize negative stain cryo-EM, negative staining electron microscopy to look them how they looks like. So if you look at the particle at the larger peak, they mostly appeared like a disk, and you can see this is a top view, and this is the side view, and this is a 2D average, okay? So if you look more into the second peak, basically pretty much it looks very similar, but it looks like a disk, but size is much smaller, and this is a 2D average. So we did measure the activity of these two peaks. As you can see here, they are both active, but with comparable activities. So as I show you, I will be showing you later, because this is a dimer of DGAT1, and based on the size as well as previous published results, we believe this population represent a tetramer of DGAT1. Okay, and for structural study, because this peak gives a higher yield and also a more homogeneity, we focus our structure study on the later peak. So we finally managed to put this peak, this protein, back to the vitreous eyes, and finally determine the qual-EM density of DGAT1 dimer at about 3.0 angstrom resolution. As you can see, this is the final density. You can see each unit has actually two DGAT1 monomers, color in different color. So, because this high resolution density map allows us to build a model very confident. So you can see this is a dimer interface, and protein from a domain swapped domain to form a dimer. And we do have some missing regions, for example, very N terminals and also very C terminals. In the middle part, few amino acid. This is probably due to those regions has a high flexibility, and we didn't resolve. So, and if you look more into DGAT1 topology respect to the ER membrane, and we can see most of the protein actually embedded in the ER membrane. And DGAT1 actually has a total 9 transmembrane domain, which are connected by loop region as well as those short helices, okay? And the dimerization interface is mainly mediated by the transmembrane number 1 as well as N-terminal soluble domain at the cytosolic phase. And N-terminal is facing to, C-terminal is facing to the ER lumen. So now we have a high resolution structure, but the long-standing question is, what is the active site of DGAT1? Okay? As it's been known for a long time, this highly conserved histidine, his 415, is a putative catalytic residues. And if we look more into this region, actually, this region is deeply embedded into the ER membrane. And besides histidine, there are several hydrophilic residues, including 2 glutamine and 1 asparagine, are also highly conserved. And also, the histidine also seems from a hydrogen bond with a nearby methionine. Okay, to verify those residues are important for the activity or not, we mutate those residues one by one to alanine and measure the protein activity. So as you can see, all of them decrease the protein activity, but 2 of them is histidine as well as this asparagine seems essential for the activity. Okay, so this collectively, those data demonstrate the active center of DGAT1 is actually embedded in the ER membrane. So now we have a good idea about the structure of DGAT1 and the active center in the membrane, then the next question is how DGAT1 capture two different substrates, okay? And then we first focus on the fatty acyl-CoA substrate. And to do this, we incubate our purified enzyme with 1 millimolar oleo-CoA, which is preferred acyl donor substrate. So in the final density map, we can see a strong additional density appeared in the structure, and this density fits pretty well to oleo-CoA molecules. Okay. And more importantly, the cell ester bond here, which is supposed to be cleaved during the catalysis, is located next to the histone, which further demonstrate here is the active center. As you can see, DGAT1 insert the oleo-CoA from the cytosolic side. This hydrophobic tail is inserted deeply into the protein and leaving this Coenzyme A head at the cytosolic site. And here is the detailed molecular interactions. As you can see, the most interaction are mediated through this phosphopentine moiety. And also the acyl chain also contribute to the interaction. However, the adenosine ring doesn't contribute at all to the interactions, okay? And we next try to mutate, select residues in this region. So we select 4 of them, and we mutate them to larger hydrophobic side chain residues, including tryptophan as well as phenylalanine. As you can see, 2 of them, this valine here, as well as this serine, if you mutate them to tryptophan, almost kills enzyme activity, which we believe this mutation probably blocks access to acyl-CoA. And we do measure the folding of each mutation we studied in our paper, and they all folded well. So that's confirmed the loss of activity is not simply due to protein misfolding. Okay. So now we have a clear picture about protein structures, active center, and where fatty acyl-CoA bind. Then what about that? And what about final react product, TG? So if we look at the DGAT1 structure again, so this is the surface cut-through view, and this is the reaction center. So the green arrow, this is the fatty acyl-CoA binding tunnel, and we do see opening led to the membrane in DGAT1, and here directly caught our attention. Why? So if we look back to DGAT1, it actually belongs to a large family called membrane-bound O-isotransferase. Okay, it exists in bacteria, in human, and in all other mammals. So at the moment, there's only one structure is available on board, which is DLTB. So this is a bacteria embodied from gram-positive bacteria. What it does is it transfers the alanine group from a protein donor, DLTC, will transfer this alanine through the also a histone embedded in the membrane to the carboxyl group at the extracellular domain called lipoteichoic acid. Okay, and to do this, this is important for cell wall biosynthesis for bacteria. So if we look at this structure and compare that with DGAT1, you can see this acyl donor binding pocket, or here is the alanine donor binding tunnel, are highly conserved, and the reaction center is in the membrane, but this gas doesn't have the opening and only present in DGAT1. Here is the cartoon representation. And if we're thinking about that, which is kind of hydrophobic, can be partitioned into the membrane, and in particular the TG product, which is highly hydrophobic. So we believe here this tunnel could be the binding site for DAG as well as the releasing pathway for the TG hydrophobic product. To kind of support this idea, actually in our either APO structure or DGAT1 complex with oleo-CoA, we do see additional lipid density at this lateral gate, which is not contributed from the protein. And this is another view, like how this density locate in the acyl-CoA complex structure. So you can see here it's located orthogonally to this acyl-CoA substrate. And if we look more into the structure, not only like two essential residues, asparagine and histidine, right next to this unknown lipid density, but all the hydrophobic residues here construct a linear pocket to house this lipid molecule, okay? And to show this portion is important for the activity, we did some mutation at this area. For example, as you can see here, this leucine 261, we mutate this to tryptophan, trying to block the tunnel. By doing that, like only 10% activity is remained. So this region is definitely important for the activity. And the linear shape of this region also explain why DGAT1 can recognize linear substrates such as DAG as well as fatty acyl fatty alcohol, but exclude the bulky, uh, potential substrate cholesterol. Actually, this isolation of cholesterol is mediated by a cat. So this two recent paper published also confirmed the binding site of cholesterol is actually also mediated through this lateral gate. Okay, and by showing all this, we are very comfortable to propose a mechanism of DGAT1. So either free fatty acyl-CoA or memory partitioned acyl-CoA can bind to this active center through this hydrophobic cytosolic tunnel. And the DAG, which is hydrophobic molecule, is bound through this lateral gate. So when both the donor and the acceptor are bound in good position, the acyl chain, by the help of histidine, is transferred to the OH group in the final in the DAG, and the final hydrophobic TG product is probably also released through this gate to the ER membranes, and the final free coenzyme A, which is soluble, is dissolved back to the cytosol. So actually, it's another same DGAT1 structure has been published, and we have very similar reaction mechanism. So to summarize, by solving DGAT1 structure, we know how the enzyme looks like, where the reaction center is, and how the enzyme captures the substrate and release the TG product. But there's still a lot of questions unknown. For example, how DGAT1 recognize different acyl-CoA or acyl acceptor substrates such as MAG, DAG, as well as fatty alcohol, and how the protein is regulated by self-organization as well as by the interaction with negative charged phospholipids. And also because DGAT1 is very important metabolic protein, a lot of inhibitors has been developed to target DGAT1, and we really want to understand how those inhibitor inhibit DGAT activities. And with that, I'd like to thank my postdoc mentor, Bob Toby and Mao Fu, for their very strong support, and they gave me really a lot of freedom to allow me explore on different things. And also, my colleagues helped me with this project and the EM facility at Harvard, UMass, and also HHMI, and finally the funding source. And with that, I'd like to take any questions. Thank you.
Thanks very much, Xuewu. So we have a few questions already. If you want, you can just type question and I'll call on you. But so John, do you have a question?
Yep. Xuewu, really great talk. I really enjoyed that. So just quick question on the dimer versus tetramer idea. So you solved the high resolution structure of the dimer, but with the 2D class averages of the tetramer, is it consistent with the gate mechanism that you're proposing, that the tetramer wouldn't block that? So I guess the question is, do you think the tetramer or the dimer, what's the more physiologically relevant oligomeric state? And then is the tetramer still consistent with that mechanism on the DAG entry?
Oh yeah, so the organization has been long proposed for the activity regulation, right? And there's several papers published before saying the tetramer is less active. Dimer is actually the physiological condition, a physiological oligomer. So in our structure, we haven't looked into this, but if you look at the ACAT structure published recently, they solve a tetramer structure. So the lateral gate is somehow blocked by the neighboring dimer. So I think that's why tetramer is lessly active, which could also be a regulation mechanism. But I think the same hypothesis also apply to BGA1, but we don't have time to investigate this yet. But there's something definitely we can do later. Yeah.
The next question is Joshua Pemberton.
Hi, great talk. In looking at the density for the acyl-CoA, I was wondering, a lot of the coordination appears to be at the top with the actual coenzyme A rather than with the extended chain. And I'm wondering if you can explain at all how the selectivity for oleoyl-CoA is being built in.
Right. So indeed, we do see like weak interaction for the acyl chain here, and the activity mostly is occurred here. So we do believe the coenzyme A portion is a major, contributor for the recognition. And the acyl chain actually, when we did the activity with a shorter length, actually the activity is much less compared with the longer chain. So we think the longer chain here could coordinate the substrate better for cleavage. But if you have a shorter chain here, this part is just like tumbling over there. So probably take a longer time for the correct position. So yes, so the major portion is actually through here. And if you look at this ACAD structure as well, and also the DCAD structure published by the other group, we pretty much see the same thing. Like here is a major contributor for the interaction. Yes, the
Okay.
acyl is probably not play a essential role for recognition. That's our understanding.
Okay, great. Thanks very much.
Sure.
The next, Forrest Bowling.
I think most of like the questions I had are kind of focused on like the tetramer potential structure and effects on biochemistry, and I think you addressed most of that with John's question. But thanks, good talk.
Okay,
So
thank
one
you.
thing is we haven't did a reduce and then more test on the tetramer thing yet, but that's something we plan to do. Probably have a different kinetics.
Okay, next question is from T. Tien Nguyen.
Yes, excellent work. I would just like to ask if you see any conformational change in the presence and absence of the two substrates that you have, and then is that a possible way to regulate when the enzyme is gonna be recruited for activity and when it's not recruited? Because you mentioned briefly about how the other monomer can block the entrance of the substrate.
Yeah. So I do have a slide, but I didn't show because for the time's sake. So overall, the protein is like rigid. We don't see like large conformational change, but we do see by compare with apo and holo structure, we do see the conformational change of this histidine. We are quite sure about that because the side chain density is very clear. So we do see like once you have the so-called bind, the histidine is like rotate to here. That's the only large conformational change we see, and this also confirms, okay, this histidine definitely is doing something during the catalysis. But other than that, we don't see anything else significant in terms of conformational change.
Next question from Grant Hatch.
Very nice talk. Enjoyed that a lot. What do you think might be the preferred substrate? Is it either a membrane-bound oleoyl-CoA, acyl-CoA, or a cytosolic acyl-CoA?
We actually has two substrates, right? If we have to think about only one substrate, which is fatty acyl-CoA. So I believe both dissolved or memory partitioned that acyl-CoA can be captured by the enzyme. And from this structure, it's really hard to say which is preferred, but my gut feeling is it also depends on the solubility, right? So my gut feeling is the acyl chain, which is more soluble, could be like a faster substrate, or the longer acyl chain is positioned better. So is really not easy to predict at this moment, and we have to do more things. Yes.
Okay, and Jinyi.
Oh, beautiful talk. And there is a previous report showing that DGAT can have a different topology, actually, topology. Can you explain this with your structure finding?
Yeah, actually, I'm very puzzled with that study in the beginning because it's really hard to imagine how one can change so significantly in its conformation. So based on our structure, because the protein is mostly hydrophobic, for me, it's really hard to believe DK1 can undergo significant, like overt or what do you call that? I forgot the name. Latent or overt conformation. So I don't think that's the case, to be honest. So I think this is the only conformation it can have to perform the function, and I don't think this large conformational chain is real. Yes.
Another question from Ronak Deo.
Hello.
Yes, we can hear you.
Yeah. Hi. Thank you for the talk. So my question was regarding the model that you've put up. The model seems to suggest that DAG enters headfirst into the protein cavity, and given like the hydrophobicity of like the middle region of the membrane, could you comment on what you're saying that?
This is kind of artificial because we at this moment we couldn't tell. The only reason I put this in the front is because this has to be goes to histone. That's the only reason I draw this like this, but it could be a totally different conformation. So it's kind of artificial. Yes.
But I would assume that that's where the active site, that's the site at which the reaction happens, right? Where the acyl chain gets transferred on. So for some reason, the molecule would have to orient itself in a way that it reaches the active site at that conformation, right?
Yes.
So would that be possible with the way that since DAG has a polar head group, and then it would have to reorient within the...
Yeah, if you look at the ACAS structure published recently, actually they got an inhibitor bound into the lateral gate, and that also mimic kind of cholesterol. And basing on that structure, I think the cholesterol always group also pointing to the histone. So I think this makes sense, but for the acyl chain, we definitely need to do something more, more to confirm that. Yes.
Okay, thank you.
I would just make one comment there that's a model. There is another potential way DAG could get in through into that pocket if it was oriented with the OH towards the bilayer towards the head groups. So it remains to be determined. We've only seen one snapshot.
So this doesn't even suggest that it's only coming from one leaflet. It could come from either leaflet in that case.
Yeah, I think we just don't have evidence. Xuewu, you can add, but I just think we don't have evidence to say for sure.
Yeah, actually, for DAG thing here, we actually don't have too much evidence, so kind of not quite confident about that. Yes.
Okay, thank you. Thanks so much.
All right, well, let's virtually thank both speakers again. Those were two really great talks. And so just as a reminder again for next week, there's no talk scheduled for next Friday. We'll be coming back in 2 weeks. and this will be posted on the ASBMB YouTube in a couple of weeks. Thanks, everybody.